Cycas introduction. 16 species of cycas are found in the tropical and subtropical parts of the earth. It is an evergreen plant. C. Revolute and C. Circinalis are most common species. Cycas revoluta is the most commonly cultivated species. It is a palm-like tree with size varies from 1 to 2 meters. As it yields sago from the mucilage in its main trunk. It is popularly called sago palm. Leaflets are 6 to 8 millimeters broad with revolute margins. Morphology. Cycas sporophyte is dioecious, i.e. male and female plants are separate and evergreen palm-like tree. The plant body is differentiated into roots, stem and leaves. Roots are of two types, i.e. normal taproot and coralloid root. Taproot is positively geotropism, non-green, without root hair. It helps in anchorage and absorbs water and minerals. Coralloid roots are lateral branches of taproot. It grows first horizontally, digotropic, then repeatedly divide dichotomously, come out of the soil surface apogeotropic in the form of coral-like mass. These are devoid of root hair and root cap. Its anatomy is similar to normal taproot, but its cortex is differentiated into three zones, i.e. upper cortex, middle cortex and lower cortex. Middle cortex carries BGA anabina cycadsurum, nostoc symbiotically. Coralloid root helps in nitrogen fixation as well as respiration due to lenticels. Stem. The stem is erect, columnar, woody and unbranched. It is covered with alternate whorls of leaf bases of foliage leaves and scale leaves. The stem apex bears a crown of leaves and sex organs at maturity. Leaves are dimorphic, i.e. two types, assimilatory or foliage leaves and scaly leaves or cataphyls. Both the leaf types are born at the stem apex in alternate spirals. Foliage leaves are large 1 to 3 meters green, pinnately compound. Petiole has 2 rows of spines. Rachis bears 80 to 100 pairs of pinnae or leaflets. Leaflets are sessile, elongated, tough, and lanceolate in shape with a spiny apex, revolute or smooth margins. Young leaves have circinate venation like those of ferns and always covered with brown hairs called ramenta. Scale leaves are small, dry, brown, triangular leaves covered with ramenta. They are non-photosynthetic and protect the stem apex. Anatomy. Normal root, young, it is circular in outline and resembles structurally with dicotyledons. Outermost layer is epibulma or exodermis, which surrounds the large parenchymatous cortex. Epibulma consists of tangentially elongated cells. From some of its cells arise root hairs. In the wide zone of parenchymatous cortex, many intercellular spaces are present. Cells of the cortex remain filled with starch. Some tannin-filled cells, mucilage cells and sometimes pharophytes are also present in the cortex. The cortex is delimited by a single-layered endodermis. Casparian strips are present in the barrel-shaped cells of the endodermis. Endodermis is followed by multilayered pericycle. Xylem and phloem bundles in the roots are radially arranged, i.e. present on different radii. The roots are usually dioic, but sometimes the number of protoxylem strands range from 3 to 8. The protoxylem consists of spiral trichades while the metaxylem consists of scalariform trichades. Vessels are absent. Phloem is present alternately with xylem groups and consists of sieve tubes and phloem parenchyma. Pith is generally absent. Young root, old root. Normal root, old, showing secondary growth. The older roots undergo secondary growth. The cambium cuts secondary phloem towards the outer side and secondary xylem towards the inner side. After some time, the pericycle cells also become meristematic and form a complete cambial ring. The secondary xylem consists of radial rows of trichades separated by parenchymatous cells. The crushed primary phloem is present in the form of dark streaks outside the secondary phloem. The secondary xylem is monoxtwalic and contains many multiseriate rays. Periderm starts to develop in the cortex of old roots. Some of the cells of the outermost cortical region start to become meristematic and function as cork cambium. Coralloid root. Anatomically, the coralloid roots resemble normal roots except some under-mentioned differences. The secondary vascular tissue in coralloid roots is either totally absent or poorly developed. The cortex is wider in comparison with the normal root. Presence of a greenish algal zone in the middle of the cortex. Algal zone consists of radially elongated, large thin-walled cells having large intercellular spaces occupied by algae. These spaces are formed because of the retardation of growth of such cells which are already infected by fungi and bacteria. Such infected cells cannot keep pace with the neighboring cells, and a tension is produced which results in the formation of air spaces by breaking of certain cells. These spaces are further widened by the algal infection. Due to the presence of blue-green algal members and some nitrogen-fixing bacteria, the function assigned to the coralloid roots is chiefly the nitrogen fixation. The presence and structure of endodermis, pericycle and vascular bundles in the coralloid roots are similar to that of normal roots. The xylem is exarch and triarch. Coralloid root stem. Stem. Similar to root, the stem of Cycas also resembles internally with a dicotyledonous stem. It shows the following anatomical features. Epidermis is the outermost layer consisting of compactly arranged thick-walled cells. Presence of several persistent leaf bases makes the epidermis a discontinuous and ruptured layer. Cortex is large and consists of thin-walled, parenchymatous cells filled densely with starch grains. It contains numerous mucilaginous canals and girdle traces. Each mucilage canal is lined with many radially elongated epithelial or secretory cells. Medullary rays connect the mucilage canals of the cortex with that of the pith. Starch in the parenchymatous cells of the cortex is the source of sago. Endodermis and pericycle are not clearly demarcated. Numerous vascular bundles remain arranged in a ring. The stellae is ectofloic siphonostele. Each vascular bundle is conjoint, collateral, open and endarch. The xylem consists of trichades and xylem parenchyma. Protoxylem contains trichades with spiral thickenings while the metaxylem has scalariform thickenings with bordered piths. Vessels are absent. The phloem is located outside the xylem and consists of sieve tubes and phloem parenchyma. Companion cells are absent. Between the xylem and phloem lies the primary cambium, which remains active only for a short period. It is soon replaced by another ring of secondary cambium somewhere in the cortex. Several broad and well-developed medullary rays are present between the vascular bundles. Pith is large, well-developed and parenchymatous. It contains many mucilaginous canals. Leaflet. Cycas leaflets are large, tough, thick and leathery. In a vertical section the leaflet is differentiated into a swollen midrib portion and two lateral wings. Epidermis is the outermost layer consisting of thick-walled cells. It is surrounded by a thick layer of cuticle. Upper epidermis is a continuous layer while the continuity of the lower epidermis is broken by many sunken stomata. On all the sides of the epidermal cells occur simple piths almost in regular series. Mesophyll is well developed and remains differentiated into palisade and spongy parenchyma. A continuous layer of palisade is present below the sclerenchymatous hypodermis. Its cells are radially elongated and filled with chloroplasts. Spongy parenchyma is present only in the wings, directly above the lower epidermis. Its cells are oval, filled with chloroplasts, and loosely arranged, having many air-filled intercellular spaces. Transfusion tissue consists of two small groups of short and wide trichade-like cells with reticulate thickenings or bordered piths on their walls. These cells have been named as transfusion tissue. Few layers of transversely elongated cells are present in both the wings just in between the palisade and spongy parenchyma. Vascular bundle is one, and present in the midrib region of the leaflet. It is conjoint, collateral, open and diploxylic. The portion of the midrib in between the palisade layer and lower hypodermal region is filled with parenchymatous cells. Some of these cells contain calcium oxalate crystals. Reproduction in Cycas. Vegetative reproduction. The most common method of vegetative propagation in Cycas is by bulbils. The bulbils develop from the axil of the scaly leaves. They are more or less oval structures with a broad base narrowing towards the apex. Several scaly leaves are arranged spirally and compactly over a dormant stem in a bulbil. On detachment from the stem, a bulbil starts germination by producing many roots towards the lower side and a leaf towards the upper side. A bulbil from male plant will develop only into the male plant, while from the female plant will form only the female plant because Cycas is a strictly dioecious plant. Sexual reproduction. Cycas is strictly dioecious, i.e. male and female sex organs are born on separate plants. After several years of vegetative growth the plants start to form sex organs. Generally, cycads of more than 10 years of age produce the sex organs. The male plants develop male cones or male strobili bearing microsporophylls, while the female plants produce a loose collection of megasporophylls. The male cone is terminal while the megasporophylls are reproduced in succession with the leaves at the top of the stem. Male reproductive structures. Male cone. The male cone or male strobilus is a large, conical or ovoid, compact, solitary and shortly stalked structure, which is generally terminal in position. It sometimes attains a length of as much as 1.5 meters. In the center of the cone is present a cone axis. Several perpendicularly attached microsporophylls are arranged around the cone axis in closely set spirals. At the base of male cone are present many young leaves. All the microsporophylls in a male cone are fertile except a few at its basal and apical parts. The terminal growth of the stem is checked for some time when a male cone appears at its apex. It is because of the fact that the apical meristem is used up during the development of the male cone. Cones of some species of cycas are amongst the largest cones in the plant kingdom. Microsporophylls, Microsporangia and microspores. Microsporophylls are flat, leaf-like, woody and brown-colored structures with narrow base and expanded upper portion. The upper expanded portion becomes pointed and is called apophyses. Narrow base is attached to the cone axis with a short stalk. Each microsporophyll contains two surfaces, i.e. an adaxial or upper surface and an abaxial or lower surface. On the adaxial surface is present a ridge-like projection in the middle and an apophyses at the apex. On the abaxial surface are present thousands of microsporangia in the middle region in the groups of 3 to 5. Each such group is called a sorus. In between these groups are present many hair-like structures, which are very soft and one or two cell structures. In tiers of a microsporophyll, there are present many microsporangia on the abaxial side. Each shortly stalked, oval or sac-like microsporangium is surrounded by 5 to 6 layers. The wall layers of each sporangium include an outer thick epidermis or exothecium, middle zone of thin wall cells and an innermost layer of tapetum. Many pollen grains or microspores are present in each sporangium. In the expanded region of microsporophyll are present many mucilaginous canals and vascular bundles. Each sporangium is provided with a radial line of dehiscence, which helps in the dispersal of spores. Each microspore or pollen grain is a rounded, unicellular and uninucleate structure surrounded by an outer thick axene and inner thin intine. Cytoplasm surrounds the centrally located nucleus. A large vacuole is also present. Female reproductive organs. Female cones. True female cone or strobilus is absent. Cycas. Female reproductive organs are present in the form of megasporophylls. Many megasporophylls are present around the apex of the monopodial trunk of the female plant above each crown of foliage and scaly leaves. Similar to foliage leaves, megasporophylls also remain spirally arranged at the apex of the stem, but their number is very large and thus they appear like a rosette. Vegetative leaves and fertile megasporophylls are produced in an alternate succession without showing any effect on apical meristem. Megasporophyll. Each megasporophyll is considered a modification of foliage leaf. It reaches up to 30 centimeters or more in length in different species. It is a flat body consisting of an upper dissected or pinnate leafy portion, middle ovule-bearing portion and proximal petiole. Petiole varies in length in different species. The middle part is comparatively wider than petiole and bears ovules arranged in two pinnate rows. The number of ovules varies from 2 to 12 in different species. The ovules are green when young, but at maturity they are fleshy and bright orange or red-colored structures. Cycas thorsi contains the largest ovule amongst the living gymnosperms, measuring about 7 centimeters in length. The megasporophylls remain covered by many yellow or brown-colored hairs. Structure of ovule. Cycas ovules are orthotropous, anatomic and shortly stalked. Generally, one or sometimes a few more ovules develop fully on a megasporophyll. Many unpollinated ones remain small and ultimately abort. The single integument is very thick and covers the ovule from all sides except a mouth-like opening called micropyle. The integument consists of 3 layers: outer, green or orange, fleshy layer called sarcotesta; middle, yellow, stony layer called sclerotesta; inner fleshy layer. Several tannin cells and mucilage canals are present in the parenchymatous region of sarcotesta. Some pigments are also present in sarcotesta and epidermis. The sclerotesta consists of linified thick wall cells. The inner fleshy layer consists of parenchymatous cells, and it remains in close association with the nucellus. The nucellus grows out into a beak-like portion called nucella beak. An enlarged megaspore or the embryo sac is present within the nucellus. The endosperm is formed by the repeated divisions of the megaspore nucleus followed by free cell formation. Just below the pollen chamber is present an archegonial chamber. 3 to 6 archegonia are present in the female gametophyte near the archegonial chamber. The latter remains filled with a fluid. Formation of megaspores. In the central region of the nucellus, the nucleus of one of the cell enlarges. Its cytoplasmic contents become dense and it also increases in its size. This cell represents the megaspore mother cell, which divides reductionally to form 4 haploid megaspores arranged in a linear tetrad. Out of these 4 megaspores, the upper 3 present towards the micropillar and the generate, leaving only the lowermost functional megaspore or embryo sac cell. This is the first cell of the female gametophyte.